Hello friends, today I'm going to make a video. I'm going to make this video on bioinformatics. This video has been done. I'm making this video with the request of one of my favorite student, and he is a bioinformatics. He's a scientist of bioinformatics, and therefore he asked me to make a video with an explanation on molecular docking. And this molecular docking is a very beautiful phenomenon. Also, you know in the students of bioinformatics and people of bioinformatics that molecular docking is a very beautiful thing, where the protein and the ligand interaction is seen and it is studied. And this process is done in many softwares in dry lab. You can make this molecular docking, and this process is very complicated. Generally, a very conventional software we use is Autodoc Vienna. It is known as Autodoc Vienna, but that process is very complicated, very lengthy and very complicated, and and therefore sometimes it fails also. In between it fails also. This error, that error, it shows different kinds of errors. So Pyrex is also there, so that also produces error. So it's very cumbersome. The process is very, very cumbersome. But there is a very beautiful software which does all this thing. Is the Discover Studio. It's a fantastic software. I should say the BioVR Discover Studio, and we is going to use this software to use this molecular docking, the featured molecular docking. Now, what is molecular docking? Molecular docking, why it is used? So, what is the actual usage of this molecular docking in bioinformatics? What information does it keep? All you know is it is a protein ligand interaction. So when a protein bonds with a ligand, or rather docks with a ligand, where it docks, how it docks, what are the properties, what are the interaction, how the amino acid behaves, these are the things which are going to study in molecular docking fission. And this is done in Discover Studio, BioVR Discover Studio, and we are going to use the version 2016. So regarding this BioVR Discover Studio, there are many confusions as I have seen in the social medias. There's a very different confusions as they say that BioVR Discover Studio, why this is the molecular docking? We cannot do. Where is the molecular docking? This Discover Studio, BioVR Discover Studio, when you download from the website, it is a free software. But all the things are free. You can prepare small molecules. You can prepare anything. Small molecules, small ligands, small proteins, even you can prepare the protein for the molecular docking, that also free. But this, you can minimize the ligand, that also free. But when you go to this molecular docking option, it is not free. It is not there. The molecular docking is not there. It requires the license. And where you will get this license, I will let you know. Where you will get the license, how will you get the license, this I will know. This I will definitely discuss with you, so don't worry. You can download it. You can whole thing, you can download it. So it's not a matter. It's not a big deal. You can download it. The thing is that you require a license, proper license file, and proper server contact. This Discover Studio is a client server method. This is a client server method. It is not just a software you just install, and with a CD key you just install and you run. No, this is not that type of software. It's a very complicated. It is a server client system. So what you have to do, you have to download the server pack, the Discover Studio, the BioVR server pack from a website that I will tell you. It is that website is known as getintopc.com. Getintopc.com. I will give all things in the description box, so don't worry. So getintopc.com. So from there you are going to get this software. So whole thing will get, it's a 6.2 GB file. It's a 6.2 GB exe file. You have to download it. You have to install in the server in your machine. It requires remember the system configuration of this software is, first of all, it should be Windows 10 only. Windows 10 64-bit. No, not Windows 11. Never go for Windows 11. Windows 11, it will not work. It will work in Windows 10 64-bit and not Windows 7. So Windows 10 64-bit and it will work. So you just install it. You get the server license, the whole thing. If you require, I will do the explanation video of that also. No, don't worry. In another way, I can discuss that also. So this is we are discussing about molecular docking. So you get that discovery pack from there, you install it, and you get that thing done. So after the installation, you will have to download two more files for your docking purpose. This docking system, you have to download two files. One is the protein file, which will be the receptor. Protein is the receptor, so we have to download a protein file from the protein data bank PDB, the PDB format, protein data bank, that RSPDB database, that RSPDB or RSPCB, that database that I will show you, that database, that file, that website also I will show you. And another one is the ligand, that is also we get from the drug bank, that drug bank database, that also that website also I will show you. From there you will download these two files in PDB format. Remember, always download this type of files in PDB format, protein data bank database, protein data bank format. So this PDB format will open in Discover Studio. Directly it will open in the Discover Studio. So after opening this Discover Studio, after downloading these files, what you are going to do, we are going to go into my desktop. We will be going to the desktop, and there we will see that how we do all this structures. And remember one thing, in Discover Studio, you need not to worry about anything. You just to download those two files. The first step is to download those two files, select two files, first of the protein and the second one is the ligand. So nothing to do with the ligand, you can leave the ligand as it is. You have to do whatever we have to do is with the protein, because protein is the receptor. So it will receive the ligand. So all the preparation is needed for the protein. So here you will have to prepare the protein. You have to prepare the active site. That all process I will show you. We have to prepare the active site. Then we have to go the docking. These are the three procedural steps. First, download the two files, protein and the ligand. Second, you have to prepare the protein. You have to prepare the active residue site, and then you have to dock with the ligand. The whole thing I will discuss in my subsystem, and I will show you the Discover Studio 2016 edition. We are going to use this 2016 edition. So let us go to the desktop. So friends, we are on the desktop, and as you can see, this is our protein files. So and this is the 6-LVL protein, and this is the phenyltephintanyl acetate. This is our ligand. So now, from where we have got this, that we are ready to discuss now. This is our site. These are the two sites. One is RCSV PDB drug data, and this is your protein. From here you will get the protein, and drug database, drug biobank online. This here you will get the drug. So that already I will give in the description box. So no need to worry about that. So now we are going to start the docking. So here we have not this one. So let us open this one. So open with Bioinformatics Studio. So this is our protein. So this is our raw protein. As you can see, this is our raw protein. So here from this receptor link, we have to go to this tab. This can open here, but this you have to go to this receptor ligand interaction. So that tab we have to use. So receptor is our protein and ligand is our ligand. So interaction. So this is your docking process. So from this tab, we are going to get that docking information. So this is our docking information. So where we are going to get all the docking systems. So now from here, we are going to define this. Here you can see a tab. It define edit and binding site. So here you have to edit and bind the site. So this from there, we have to first add the hydrogen, and from this we are going to prepare the ligand. We have to first prepare the ligand. You can see all the ligand groups, this protein and these things. You are here from this. You just go to this arrow, this small arrow you can see here, the small arrow. Just start this arrow and you will get all these things. So here we are seeing the ligands already. The two ligands are present, ligand 1 and ligand 2 are present. So from here we are going to prepare the active site. Now we have to prepare the site, because where the ligand will join or the bond, or rather dock, so that you have to define. So we are going to define only one binding site. Two binding sites, two ligands are there. So we are going to delete one ligand. So select one ligand, So to select this ligand and we're going to delete and we'll keep this ligand. This was ligand 1 we will keep. So let us select this ligand, and from this current selection we are going to active site. So here is the active site. So already the active site has been done, and now we can delete the ligand, because one ligand if it is present, then another ligand will not talk, definitely not talk. So now this is our prepared protein, and we can save as 6LVL and underscore prepared. So our protein is prepared. So our protein is ready over here. Now we have to open the ligand. Open the ligand. So from here we are going to open this phentenyl acid site. So this is our ligand is done. So this is our protein and our ligand. So both are ready now. Ligand you have to do nothing. Ligand you need not to do anything. So we are all going to with the protein here. So our site is ready, and now we are going to talk the ligand. So here is our lip talk, and this is our talking link talk. This is our option. So this we are going to use. So lip talk, and now the is open. This lip talk site has been opened, and you can see the input receptor. What we can see here, we see the input receptor, that is our protein, prepared protein, 6LVL protein. Now this is our ligand. So ligand, it should not be protein, it should be our phentenyl cited, which is visible here. As you can see, this is phentenyl cited visible. So this should be there. Visible should be visible. It should not be invisible. It should be visible. And be careful, don't make this receptor as phentenyl cited. Otherwise, your input receptor will go. So that you cannot do. You have to use this receptor. So receptor is protein only. Now you can see the input sphere, binding sphere, sphere coordinates are here, the docking tolerance is here. So here you can minimize the ligands, you can minimize the ligands, you can do anything. So we are not going to minimize the ligand. So we are just going to keep it, this for demonstration purpose, we are going to keep it. Otherwise you can do anything, any information. So that you can do, it is your choice. So now we are going to run the docking, and you can see the details. Now, first of all, it is preparing the protein, and then it is preparing the ligand. Now it is preparing the protein. The first step, you can see the 3 steps. So first step is preparing the ligand. The phentenyl cited, it has already prepared the ligand. Now it is preparing the protein. So this will take time. So this usually takes time. So this you will have to bear, and this will take time. So the work is going on, and you can see it is prepared, and so it is preparing, and it will taking time, little bit take time, and you can do bear in patience. So already the docking has been done, and you can see the docking sites over here. Now you can see the docking over here, and just if you go to the zoom, and you can see the docking, it is here inside the sphere. You can see the ligand has been docked. So you can see more positions, and here one thing you can go to the view interaction. So here you can see the interactions, and you see the ligand interactions. So this is your ligand interactions, it's been done. So this is interaction has been done. So different positions are there. 100 positions are there. So 99 positions are there, and you can see any position just by clicking the mouse. And here in the bottom, you can see the amino acids. You can see the non-bond anything, and all you can see here. So all interactions are there, interactions, and you want to expand the interaction option, you can expand, and you can just see the whole thing, or you can contract, or you can do anything with this. So you can go to the structure, and you can see the structure in 2D, the ligand structure of ligand in 2D. So this is the 3D, and you can see the structure in 2D. So this is all about your docking procedure in Discovery Studio. So thank you very much.